Thank you for the introduction and welcome everybody. It's nice to see so many young people interested in science in this room. Actually, Professor Bari was supposed to give this talk. He submitted the abstract, but he is needed in Debrecen today, so I'm going to present his research interest. As the title indicated, we are very much interested in acute brain injury. There are many types of acute brain injury. Probably the most known type of acute brain injury is traumatic brain injury, when somebody hits their head. But stroke is also another type of acute brain injury, acute because it has a very sudden onset. Patients who suffer of this disease probably get a headache or get dizzy, sometimes collapse or lose consciousness all of a sudden. And the symptoms of the disease are quite typical, but oftentimes are not recognized. A very clear symptom of the disease is when one side of the patient's face is dropping. Oftentimes, when there is a suspicion that the patient has a stroke attack, the patient is asked to smile. And these people can smile only with one side of their face, like this lady in this picture, and the other side remains immobile. Oftentimes, they cannot move their arm or one limb on one side of the body. And one test to see if this is the case is to ask the patient to raise their hands or arms in front. And if there is a stroke going on, then this patient is not going to be able to keep the two arms level, but one arm is going to drop against the patient's will. What's going on in the brain when all these symptoms occur? There are two options, because stroke has two types. One is what we call ischemic stroke, when one blood vessel in the brain, which supplies a certain brain region, gets occluded. And because of that occlusion, blood flow, blood supply to that particular brain area ceases. Another possibility is that a blood vessel in the brain gets ruptured and a hemorrhagic stroke occurs, which means that there is bleeding inside the brain tissue. Most of stroke cases, maybe 70–80% of the cases, are ischemic, so they are caused by a sudden blockade of a blood vessel. And in 20–30% of the cases, there is a rupture behind the symptoms, and the two must be discriminated when the patient is admitted to hospital because they require different treatment. This disease, stroke, is not selective to people. Anyone can get a stroke, as these famous examples demonstrate. And if you look around in this room, try to imagine that from every 6 person, one will get stroke sometime, probably later during their lifetime. I have a higher chance to get a stroke than you do because I'm 30 years older than you are, and aging is a very significant risk factor to get a stroke. The outcome may be variable. For Charles Dickens, this stroke attack was deadly because he died the day after his stroke attack. Sharon Stone is said to be cured, so the outcome is probably dependent on the severity of the stroke attack and also on the quality of the health care the patients receive. Okay, so we are interested in this devastating disease because, first of all, we would like to understand what causes cell damage in the brain when stroke occurs. And second, we would like to find ways to limit that cell damage, if there is any way to improve the patient's condition. It is a very challenging demand because in order to help a stroke patient, the patient must get into hospital within 4 hours. It's a very, very short time window for intervention. And for that reason, Secondary injury may be in the focus of therapy. Secondary injury is something that takes place days, weeks after the stroke attack, and these secondary injuries worsen the condition of the patient. So this is also valid aim and plausible aim to reduce secondary neurodegeneration, which was in the title of this talk. Okay, first question. We would like to understand what causes cell damage. And one condition which is supposed to cause cell damage is tissue pH decrease, acidosis in the nervous tissue. Here I show you a neuron. This neuron is in a culture, in a dish. The picture was taken with electron microscopy, and this neuron was cultured at a pH of 7.4, which is quite normal, and this cell looks quite normal. If we would like to look at the mitochondria, which are these dark potatoes in the image, we can see a very nice preserved structure of these organelles within the neuron. Now let's change the pH of the medium to an acidic pH 6. Now, in this case, the appearance of a neuron is swollen. That is already an indication that something is going on, something is wrong with this cell. And if we look at the mitochondria of this neuron, we can see that these are swollen bubbles without clear-cut structure within them. And this is also a clear indication that the cell is dying. So this is something which is quite bad. Now, because under these conditions in the dish, tissue pH is the only thing manipulated, we can draw the conclusion that acidosis is really killing those cells. How? It may kill those cells because acidosis leads to the generation of free radicals, which will damage cell membranes and DNA and peptides as well. Acidosis will disrupt signaling within these cells, so they cannot operate properly. And the DNA within the cell nucleus gets fragmented as well. There are certain ion channels, acid-sensing ion channels on cell membranes of neurons, which can also signal death pathways within the neurons, and therefore the neurons will die. In our lab, we decided, or we wanted to monitor acidosis in our stroke models. So we came up with an imaging solution in which anesthetized rodents were the subject of this investigation. We created a window in the skull of these animals so we could look at the brain surface, and we loaded the cortical tissue with a dye, a fluorescent dye. And this fluorescent dye is very specific because it increases the intensity of the fluorescence when tissue pH is decreasing. So it increases the optical signal when we have acidosis going on in the tissue. Together with monitoring tissue pH, we also can look at blood flow variations in the same area. This technology is based on the use of lasers, and the blood flow maps are created on the basis of the reflectance of the laser, which gives a pattern. And we can create these very nice colorful images in which warm colors, red indicates high flow areas, and cool colors like blue and green indicate low flow areas. Now, what happens if we cause a stroke by occluding a blood vessel in such an animal? Well, in the pH image you don't see much yet, but within 25 seconds, blood flow in this area very dramatically drops to 30 to 40% of the original flow rate, which can be visualized by the color change. Now, in the pH image, the intensity, the red color intensity would increase, would indicate acidosis, and if I show you the subsequent images, you can very nicely follow the evolution of acidosis in the tissue, so we can visualize decreasing pH by this method. And we can confirm that in our stroke model, acidosis is a real phenomenon. It goes on not only in a dish, like in the previous example, but in the living organism as well. Later on, I will come back to acidosis, but right now I would like to switch to the other aim of our research, namely to find ways to counteract cell damage that occurs during stroke. We can visualize cell damage in rodents, in mice and rats, by slicing their brain and coloring the brain slices for living tissue. The dye which gives this red color is called tetrazonium chloride, and this dye labels viable tissue. Now, these slices come from an intact animal, and the next series of slices come from an animal which had a unilateral stroke, and at that side of the brain, the color does not evolve because this tissue is dead. So with the help of this technology, we can estimate the size of the infarct caused by vessel occlusion. Now, what happens if we treat these slices or the animals with a drug called nimodipine? Nimodipine is a drug which dilates blood vessels, so it will increase perfusion. And it also helps neurons survive during any insult. Okay, so we treat the mice with nimodipine and look at the infarct size again. We see that in the treated animals, the infarct size is smaller. So this drug is quite useful. We can quantitate the change by measuring the ratio of the infarcted area compared to the whole size of the brain slice. And we can see that in an animal with a stroke, there is a large proportion of infarcted tissue, but in an animal with nimodipine treatment, we can decrease the size of the infarct lower than 50%. This is nice, but all benefit comes at a cost. And the cost in this case, which I demonstrate with a trace from patients under surgery, the cost is that this drug, nimodipine, because it dilates blood vessels, it causes hypotension. Hypotension means that the reduction of blood pressure. And the reduction of blood pressure leads to low perfusion pressure in the brain. And if there is low perfusion pressure, there is low perfusion, and that is really bad to a brain which suffers of stroke. So we would like to keep the beneficial effect, but we would like to get rid of the side effect, which is not good for the patient. And this is a challenge, which is not easy to obtain. And now, I would like to show what solution we have come up with or trying to test. If you attended the morning session, one of the speakers, Gergő Porkoláb, talked about nanoparticles. Now, nanoparticles are very nice particles, a very nice way for drug delivery. Gergő told you that nanoparticles are useful because the blood-brain barrier is not penetrable, and with the help of nanoparticles, you can get drugs into the brain. Now, we had a different idea because we would like to use nanoparticles to deliver nimodipine, the drug previously shown, specifically to the injured brain region and nowhere else. We didn't want to see vasodilation or hypotension. Therefore, we approached the group of Prof. Imre Dékány and László Janovák and asked them whether they could manufacture nanoparticles for us which are sensitive to pH, tissue pH. Why? Because, as I have shown you, tissue pH drops where there is an injury going on, so this would be a good indicator of injury and a good signal for drug release. Okay. Now, they have created nanoparticles which are made of chitosan. Chitosan is a derivative of chitin, and chitin is best known to be the element of the exoskeleton of insects, and it can be also found in the cell wall of mushrooms. So it is a biocompatible compound, and it gets degraded in the body and gets cleared away, so we can use such a molecule without much risk that the patient will develop infection or an inflammatory reaction to the nanoparticle. The size is very small. It's about 5 nanometers. It's really, really small. So we associated nimodipine to these particles and looked whether these particles really respond to pH and release nimodipine at low pH. Well, first, the pH profile was determined in suspension, so not in a living organism. And as you can see, this would be the suspension of the nanoparticles at physiologically normal pH. And as pH decreases, the nanoparticles change and they release nimodipine. And the nimodipine release starts at around pH 7, and it's most effective at pH 6.5. All right, so we watched this suspension on the surface of the brain of anesthetized rats, which were subjected to ischemia, brain ischemia or stroke. Now, this graph may be complicated, but it's a very important plot. The gray bars, stand for tissue pH, which we can measure with pH-sensitive microelectrodes. And this part of the experiment, the first 2 bars, are the control period where tissue pH is normal. Then at this point, we occlude a blood vessel which supplies the brain with blood. The circles stand for cerebral blood flow, and at that point, cerebral blood flow drops very suddenly in 2 groups of animals, one which is treated with nanoparticles alone, and one which is treated with nanoparticles carrying nimodipine. At that point, the gray bar tissue pH also drops, and this would be the signal for the nanoparticle to release nimodipine. Okay, let's see what happens at the next sampling time. At this point, the 2 groups, one with nimodipine and the other one without, start to be different. The one with nimodipine has an improved cerebral blood flow, and the other one has a similar level of blood flow, which means indirectly that by this pH drop here, the nanoparticles release nimodipine, and nimodipine dilates blood vessels at the area of interest in the brain. So this approach seems to work, at least under these experimental conditions. Then we went on to see whether we can achieve neuroprotection. And these are brain slices labeled for neurons. So the black or dark dots are indicating neurons, stand for neurons. In this slice, you see a pale area up here. This is the cortical surface. And this animal received nanoparticles only, no nimodipine. So we have a cell loss in the upper layers of the cortex. Let's see what happens when nanoparticles carried nimodipine to the brain surface. The integrity of the cortex looks normal. There is no conspicuous cell loss in this representative animal. So by the nanoparticles which carried nimodipine, we could increase blood flow in the cortex after a stroke, and we could protect the neurons as well. Now we need to move on and find out how to apply these nanoparticles to the animals in such a way that we can try to do it in patients as well, but that's the future perspective of this research activity. Now, who are the people behind this work? I'm always proud to show you the group of cerebral blood flow and metabolism, which is a very nice group of people conducting this sort of research at the Department of Medical Physics and Informatics. If we zoom in to see the people involved in the STA, the Szeged Scientists Academy, in this research group, Professor Bari and myself are mentors, and Dr. Ákos Menyhárt and Daniel Varga are junior mentors. And at the moment, we have 3 STA students, Dóra Hantosi, Réka Tóth, and Armand Bálint. And as you can see, this is a quite successful team. Where to find us? Well, we are at the riverbank. If you are familiar with the buildings there, and you know where the eye clinic, the clinic of ophthalmology is, and the internal medicine clinic, our small building is right in between these two. And we have research laboratories, almost 5 research laboratories in this building. Within the STA, the most important international collaboration we have is with the group of Professor Zoltán Ungvári in the United States at the University of Oklahoma. He is an international mentor in the STA program, and his main research profile is the aging of the brain, with particular attention to the blood vessels in the brain. And Anna Csiszár is his wife and co-worker. We have also other international collaborations because our research is embedded in the efforts of the COSBIT group, which is the Cooperative Studies on Brain Injury Depolarizations. And we have intensive connections with Dr. Stefan Marinescu in France, Lyon, and Jens Dreyer in Berlin, Germany. Stefan is not so severe, but this is his profile picture. He's a warm-hearted person. And by that, thank you very much for listening to this talk, and I'm open for any questions you might have. If you would like to know more about us, these are the email addresses through which you can reach either Professor Barry or myself. Thank you very much.